Ed, the best introduction ever, and a big thank you to Ashir, Fran, and Felix for the chance to be here today. And I'll not be quite so erudite in my Harry Potter puns throughout the talk. I'll keep them to a minimum. But I'm going to be explaining to you today a story that we've assembled over the last few years on protein-lipid phase coupling, and how it's involved in lysosomal and RNA transport, primarily in neurons. And I guess this isn't a classical membrane trafficking talk in the conventional sense, but I hope that it involves membranes and trafficking. So I hope that you'll find it interesting still. So, okay, I often like to start by showing this movie, which is actually a pretty standard fibroblast cell with a bunch of different fluorescently labeled organelles. But I really like this movie because I think it highlights, first of all, just how busy and dynamic a place the organella landscape is, and I think really helps to illustrate the pretty remarkable frequency with which organelles sort of encounter each other across the entirety of the cell. And as I think most people in this audience will be aware of, we now know that these organelle contacts, these organelle encounters, aren't simply arising as a product of chance, and actually they represent a very tightly regulated and critical feature of eukaryotic cell biology. And so these direct interactions between organelles in the form of contact sites have now been established between pretty much every intracellular organelle and across almost all cell types. And in terms of their functions, we know that contact sites play a really key role in mediating the exchange of a number of different biomolecules between organelles, but also, and this is really going to be the focus of my talk today, they play a very important role in dictating the size, the morphology, and the position of several organelles. And I know that many people in this audience will be familiar with this in the context of, for example, ER mitochondria or ER endosomal contact sites templating locations for organelle fission. But actually, the story that I'm going to talk to you about today is an example of how contact sites can tether organelles together for co-trafficking purposes. So what do I mean when I'm talking about tethering here? Well, the formation and the disassembly of contact sites is it's a tightly regulated process coordinated by very specialized protein tethering machinery. And one of the things that we're interested in my lab is understanding how this tethering machinery works, and as Ishir alluded to, how it's then misregulated in the context of disease. So a lot of the work that we and many other groups have focused on have been geared towards understanding tethering machinery acting between two membrane-bound organelles. And so you can see here a very well characterized example of that, where this protein VAT-B acts to tether ER to mitochondria. But what I want to really focus on today is a contact site between a membrane-bound and a membrane-less compartment. So between the lysosome, of course, membrane-bound, and the ribonucleic protein granule, a membrane-less organelle that I'll go on to talk about. And I'm going to tell you about this pretty unique tether, an XNA11, that we think can act to essentially couple the biophysical states of both proteins and lipids in this system to ultimately facilitate co-trafficking of both of these organelles. But before I get into the meat of that, I need to give you a bit of background on how we came across an XNA11 in the first place and why we think it's important. And so I'm going to tell you very briefly about some work that we were involved in a few years ago that really was spearheaded by Yacheng Liao, who at the time was in the Lippincott-Schwartz lab, is now a group leader at Columbia, and Michael Fernandopulle, who at the time was in Michael Ward's lab at the NIH and has now gone on to do his residency. He's in fact just applied for residency now. He's a fantastic and brilliant young scientist. So keep your eyes out for him in the future. Returns to research. And so their work really focused on understanding a simple but long-standing question on how mRNA is trafficked. And so if you think about this problem in most cell types, RNA is transcribed in the nucleus, and then if the cell needs a protein, for example, to be synthesized near the periphery of the cell, it can do this relatively easy. You're talking about a distance of the mRNA needing to travel of something in the order of 100 microns. When it comes to thinking about extremely polarized cells like neurons, then this distance can extend to, upwards of a meter long. And we know that mRNA reaches these sort of distal portions of neurons because we can see it there. And I think there's pretty compelling evidence from a number of labs, Erin Schumann, Tom Schwartz, and so on, demonstrating quite clearly that these mRNA are being actively translated at these distal sites. And in fact, we can even see labeled mRNAs moving up and down axons, and various other groups have shown that these mRNAs don't travel on their own, but they form part of membraneless compartments that I mentioned before, called RNP granules, or ribonucleic protein granules, which, as the name suggests, are a complex mix of RNAs and proteins that we'll get on to more detail shortly. But this really begs the question, of course, the basic question: how do these RNP granules actually traffic down axons? So we know very well in the case of sort of membrane-bound organelles that they can traffic down neurons by engaging with motor proteins, which then drag them up and down microtubules, like you can see here in this example of a lysosome. But we also know that this isn't so simple for RNP granules, which in many cases, not all cases, but in many cases can't directly engage with motor proteins. And so to cut a very long story short of many years of work, what Yacheng and Michael were able to demonstrate is that RNP granules could attach themselves to lysosomes in order to hitchhike a ride down axons. And they were able to identify an XNA11 as a key molecular tether linking the lysosome and the RNP granule together for these hitchhiking purposes. And you can see, I think, a really nice example of that here, where essentially you're looking at a single axon. And I hope what you can appreciate is that these sort of pronounced co-trafficking events, where we see lysosomes and XNA11 and RNP granules co-trafficking together. Okay, so shifting gears a little bit, I've talked sort of vaguely about RNP granules being membraneless compartments, but what do I exactly mean by this? Well, RNP granules are a broader part of a broader family of membraneless structures inside cells called biomolecular condensates, which I imagine everybody here now is extremely well familiar with. But just to recap some basics, a really important defining characteristic of condensates is that they're formed by phase transitions. And we now have pretty good evidence that low sequence complexity regions within some proteins allow them to undergo these phase transitions. And what you're looking at here is one of these low complexity, or LCD, containing proteins purified in vitro, as it transitions from this dispersed state into this condensed phase, and then ultimately into a gel-like phase, which we can determine by assessing the relative fluidity of the protein. As I mentioned, it's these types of phase transitions that are thought to underpin the formation of membraneless biomolecular condensates like RNP granules. But what some of you may have noticed from the image in the middle is that actually many of these condensates are found in close proximity to, or actually even directly tethered to, lipid membranes. And this was very interesting to us, given there's also, again, as many people will be aware of, a significant body of work demonstrating the importance of phase transitions in lipids. And you can see that represented in this cartoon here, where essentially the lipids transition from a disordered liquid-like phase, where the lipids are mobile and free to diffuse, to a gelled phase, or an ordered phase, where the lipids are tightly packed and immobilized. And this change in fluidity and ordering of lipids has been shown to have very profound effects on the overall mechanical properties and, of course, organization of various membranes, both at the edge of the cell, the plasma membrane, and also throughout cells. So we can have both protein and lipid phase transitions occurring inside cells. And so coming back to an XNA11, I'm going to show you a really highly specialized molecular tether that can mediate both of these processes. And what we found is that it does this through a pretty unique domain architecture of two distinct domains, where at its N-terminus, an exon has a low complexity domain, which we've shown allows the protein to undergo a concentration-dependent phase transition from a dispersed to a condensed phase, so no crowding agents or anything here. And then when we remove the low complexity domain, that impairs the ability of an exon to form condensates. And it's this phase transition that's very important for its interaction and its ability to engage with RNP granules. Then at the C-terminus of the protein, we have this series of annexin repeat domains, which we've shown essentially allows annexin to bind to lysosomes. And again, when we remove this series of annexin repeat domains, this binding to lysosomes no longer occurs. And really crucially, what we've then been able to show is that this binding of annexin to lysosomes causes a gel-like phase transition in the underlying lipids of the lysosomal membrane, sort of stiffening them up. And you can see one example of that shown here with a optical reporter that reads out lipid phase states. I'm going to go on to discuss that in more detail in a minute. But the ARD alone, just binding of the ARD, is sufficient to induce this gel-like state change in the lipids. But what we really wondered then was whether annexin might be able to sort of act to couple protein and lipid phase states such that it could transfer information about the protein phase through to the lipid phase. And this, I think, would provide a sort of important conceptual link between lipid membrane properties and biomolecular condensates, which are two, of course, key principles governing cellular organization, but also in the very specific context of the lysosome RNP granule system, we thought that perhaps phase coupling might play an important role in the capture and release of RNP granules at specific locations or sites in neurons. And so to test this, we decided to build a simple in vitro reconstitution system. And so for our lipid component here, we generated giant unilamellar vesicles, GUVs, with a lipid composition that very closely matched that of lysosomes. And importantly here, it includes the phospholipid PI3P, which is the major lipid that annexin binds to. What we could then do is add physiological concentrations of fluorescently labeled purified annexin A11 to our GUVs, which binds to the membrane in the presence of calcium. And you can see that shown here with annexin shown in red being recruited to the membrane surface. So to then test our idea of phase coupling using this reconstitution system, what we want to be able to do is change the protein phase state of annexin A11 and then see if we can detect a coupled change in the phase state of the underlying lipids in our lysosomal-like membrane. And typically, the easiest way to induce a change in a protein phase state in an in vitro system like this would be by altering a physical or chemical parameter like temperature, crowding, changing the salt concentration. But the problem here is that these factors are also going to directly impact the lipid phase state. And so that's an issue. And of course, these parameters also are not necessarily the most physiologically relevant. So instead, what we decided to do was to take a slightly different approach. And so what we did was start by screening a number of proteins which we identified as annexin A11 interactors for a couple of key attributes. Firstly, whether or not they could alter the phase state of annexin A11, and then secondly, that they didn't directly modulate the lipid phase state. And I'll show you how we did that now in the context of two, I think, particularly interesting interactors, ALG2 and calcyclin. Both of these two proteins are small, soluble cytosolic proteins that interact with a small structured motif in the annexin A11 low complexity domain. Okay, so firstly, to look at the impact of the interactors on the phase state of annexin, we can look at the mobility of annexin on GUVs in the presence of ALG2 or calcyclin using FRAP, so fluorescence recovery after photobleaching. And essentially what this data tells us is that annexin is pushed into a more immobile or gel-like phase in the presence of ALG2, and in the opposite direction into a more mobile or fluid phase in the presence of calcyclin. And I'm not going to go into the data here today, but I think it's very important to mention that we also checked with FTIR, and the effects of ALG2 here on annexin don't correspond with an increase in intermolecular beta sheet content, suggesting that this is a sort of phase transition that is not occurring as a result of classical aggregation. So as I mentioned, we then needed to go ahead and check whether ALG2 and calcyclin on their own, without annexin, had any effect on the lipid phase state. And so to do this, we used a PK dye, which I referenced a few minutes ago, which is a solvatochromic dye developed by Andrei Klimchenko's group that essentially intercalates into lipid membranes and changes its fluorescence based on the lipid phase state. And you can see here the PK dye in action. So the top panels just show the GUV membrane, and then on the bottom panels, you see the PK dye readout of the change in lipid phase state. And as you can see, the co-incubation of our lysosomal-like membranes with either ALG2 or calcyclin, again, on their own, no annexin present, has no effect on the lipid phase state. So collectively, then, I think ALG2 and calcyclin, we reason, provided us with a pretty powerful and physiologically relevant method to tune the phase state of an XNA11 to be either more liquid in the case of calcyclin or more gelled in the case of ALG2, but crucially, without having any direct effects on the phase state of our lipid membranes. So we can now then use these two modulator proteins to test our idea of phase coupling. And we do this in a very simple experiment where we take our lysosomal-like membranes, which we've preloaded with annexin and calcium. So the annexin's recruited to the membrane surface. And we can then add into this our two modulator proteins to alter annexin's phase state and read out the coupled changes in the underlying lipid using our PK dye. And so what you're looking at here first is just an annexin-coated GUV and its relative lipid phase state. When we then add ALG2 into this system, so remember driving annexin into a more gel-like state, you can see that this has this really dramatic sort of coupled effect on the underlying lipids, causing them to stiffen up and be pushed into a more ordered or gel-like state. In contrast, when we make annexin more fluid by adding calcyclin, we can see that this results in a coupled change in the lipids where they also become more fluid, which you can again see shown really clearly here in the PK dye. And in the interest of time, again, I won't go into the data today, but these PK dye measurements that we've taken are also corroborated by other orthogonal approaches. So we did some lipid FRAP and some nano AFM infrared spectroscopy to look at the phase states of both the proteins and the lipids, and the changes that we see in the lipids that I'm showing you here with the PK dye were mirrored with both of those two approaches. So this was pretty cool. We were excited about this, but we wanted to make sure that what we were seeing in vitro held true inside cells. And so to do this, we exploited the fact that the PK dye we're using was cell permeable, and we used it to extract the lipid phase state of lysosomes here in living cells. And as you can see, very similarly to our in vitro system, if we co-express an annexin with ALG2, this results in more gel-like lysosomal membranes or more ordered lysosomal membranes. Whereas when we co-express with calcyclin, this pushes the membranes into a more fluid state. Again, co-expression of each of these two, single expression of each of these two proteins alone didn't have quite the same effect. Okay, so taken together, I think this data for us painted a pretty compelling picture of an XNA11 as a new class of molecular tether, really, that's capable of coupling protein and lipid phase state changes. And the next question that we wanted to ask was, how does this relate to annexin's ability to tether RNP granules? So to test this, we essentially purified RNP granules using a FAPS-based approach, a fluorescence-activated particle sorting-based approach, purified them directly from cells, and then added them into our in vitro system, where we could then essentially quantitate their binding to annexin-coated lysosomal membranes. We then dumped ALG2 or calcyclin into the system to again change the annexin phase state, and we see that stiffening of the annexin lysosome ensemble with ALG2 corresponded with more RNP granule binding. And if we fluidize the ensemble, softening it up with calcyclin, this decreased RNP granule binding. And ultimately, this is speculative, but what we think is that this altered tethering might play an important role in the trafficking of RNP granules down axons by modulating their capture and release from lysosomes at sort of specific pickup and drop-off locations. And as I mentioned at the start of the talk, having RNP granules picked up and dropped off at the right places is super important in ensuring the delivery of mRNA payloads to the right locations to enable local protein synthesis and ultimately ensure neuronal function and viability. But when we think about RNP trafficking, there's also another important stage to consider beyond the pickup and drop-off locations, and that's what's happening during the actual trafficking phase itself. And this is interesting to us because we know that there are pretty considerable shear stresses applied to cargoes when they're trafficking down axons. There are variable measurements of this, but a ballpark figure would be 100 piconewtons per micron squared. And so what we wondered was whether the phase coupling phenomenon that we've identified might alter the nanomechanical properties of the lysosome RNP and annexin ensemble to make them more resistant to these shear stresses. And so to look at this, we collaborated with Yi Shen, who's at the time a very talented postdoc in the Knowles lab in the chemistry department here in Cambridge, has since moved on to set up her own group in Australia. And she built a microfluidic device that essentially forces GUVs into these tapered glass tubes of decreasing diameters to simulate different shear stresses. And from this, she was able to extract the elastic modulus of GUVs on their own and in the presence of an XNA11 with either ALG2 or calcyclin. And the elastic modulus, or Young's modulus here, essentially gives us a readout of the stiffness of the ensemble. And from this data, you can see quite clearly that having an XNA11 on the surface of lipid membranes substantially increases the stiffness of the ensemble. And this increase in stiffness is enhanced even further in the presence of ALG2 and decreased in the presence of calcyclin. It's worth mentioning again that we've tested ALG2 and calcyclin on their own without an XNA11, and they have no effect on the GUV stiffnesses. So again, of course, this is speculative, but perhaps phase coupling here might also act to modulate the nanomechanical properties of the ensemble to sort of resist these shear stresses associated with axonal transport. Okay, so wrapping up, I've talked to you today about an XNA11, this pretty unusual tethering protein that uses its specialized domain architecture to alter both protein and lipid phase states. What we've been able to show is that by modulating the phase state of an XNA11 is capable of transferring information about its protein phase through to the underlying lipid membranes through a phase coupling mechanism. I then went on to show you that this phase coupling correlates with the capacity of the annexin lysosome ensemble to bind RNP granules, which we think may play an important role in the control of their capture and release sites within neurons, and also that the overall nanomechanical stiffness of the ensemble can be modulated by phase coupling, which again, speculatively, might provide resistance to shear stresses during trafficking. So I've focused, of course, today on the lysosome RNP granule, but as I mentioned, there's a bunch of other condensates in close proximity to membranes. So I think we're really just scratching the surface of this phenomenon here. And actually, there are a number of other groups now that have published some fantastic work looking at some of these other condensate membrane systems. So I think, you know, phase coupling is very likely a sort of general organizational mechanism that applies to multiple scenarios across the cell. So with that, I'll wrap up and just say a big thanks to everyone in the Hislop lab, in particular Seema, who did a lot of the work purifying the proteins, collaborators in the Knowles group down in chemistry here. I mentioned Yi Shen, but also Simone Ruggieri, who did a lot of the nano-IR work that I haven't shown you here today, and then various collaborators dotted around the world. And of course, I mentioned Michael and Yacheng's role in the original work identifying an exon as this tether. With that, I'll wrap up and happy to take any questions.